Hi everyone, thanks for joining today. We'll get started in 5 minutes. Alrighty, let's go ahead and get started. Hi everyone, thank you for joining today and welcome to the NOAA Omics seminar series. I'm your host, Nicole Miller, and I sit in NOAA Ocean Exploration. Omics, which is a suite of tools used to analyze DNA, RNA, proteins, and metabolites, has become a large priority here at NOAA in the last few years. We established this seminar series in an effort to increase transparency and collaboration and highlight the incredible omics-related research currently underway both within and outside NOAA. The seminars from this series are made available on YouTube and posted on our NOAA Omics website. There should be a link to the website in the chat now. As participants, you are muted, but feel free to type questions in the questions for staff box throughout the presentation, And at the end, I will read the questions for the presenter to answer. So with that, our presentation today is titled Great Lakes Atlas of Multi-Omics Research, or GLAMR, by Dr. Anders Kiledal. I'll go ahead and allow sharing. Anders is an assistant research scientist at the University of Michigan, where he leads development of the Great Lakes Atlas of Multi-Omics Research, or GLAMR, and uses omics techniques to study harmful algal blooms, particularly the large annual blooms that plague Lake Erie's western basin. He received his PhD from the University of Delaware, where he used omics techniques to study bacteria living in and on concrete. Thanks, Anders, and I'll now pass it off to you.
Great, thank you so much, Nicole, and thanks to everyone who's tuning in live or checking this recording out on the NOAA Omics Seminar Series YouTube page. So like Nicole said, my name is Anders Kiledal, and I'm a research track faculty member in the Earth and Environmental Sciences Department here at the University of Michigan. So today I'm excited to tell you about a platform we're building to more easily find, analyze, and contextualize omics data sets from the Great Lakes. This project has been supported by the NOAA Omics program via the NOAA Great Lakes Environmental Research Laboratory, and I first joined this effort as a postdoc working with Greg Dick, who also directs the Cooperative Institute for Great Lakes Research. However, as you can see from this title slide, there are many people who've been instrumental to this effort. It's great to have an audience that probably doesn't require too much of an introduction to omics methods and the ways they've been changing how we study ecosystems and aquatic ecosystems in particular. But nevertheless, these omics techniques refer to several high throughput technologies that allow for the characterization and quantification of the pool of macromolecules in an environment. So these macromolecules can include DNA, RNA, proteins, and metabolites, and they contain a wealth of information about who is present in the community, such as some of these organisms you might find in the western basin of Lake Erie, the genes that they're expressing, so from RNA data, and this can also reveal information about the stressors they're experiencing or the compounds they're potentially producing. So as this technology has improved, particularly over the last 20 years, it's ushered in new and powerful ways of studying the environment and its ecosystems. And of particular interest to many in the Great Lakes, omics techniques are providing expanded monitoring capabilities and valuable insights into the biology of harmful algal blooms. So while these techniques are already having a significant improvement or impact on our understanding of the biology, they also hold a lot of potential for informing next generation modeling, forecasting, and management practices. So currently, a lot of studies produce these individual data sets, but by integrating them, there are these opportunities for, say, meta-analyses or bioprospecting or next generation monitoring. And by producing standardized data products of these really integrated data sets, and also standardizing the metadata associated with them, they present many opportunities for modeling, forecasting, and management. So before GLAMR, we know that the data is out there. It's being deposited, for instance, in public repositories like NCBI, SRA, or the Sequence Read Archive. However, if you've ever had the displeasure of trying to track down data from SRA or potentially related databases, you know that sometimes the metadata is really lacking and certainly not standardized. So here's an example of a somewhat random sample chosen from SRA. And you can see, okay, we have the location, Lake Erie, even some GPS coordinates, but there's no other information about, say, when this sample was collected or any other context associated with it. Add to that, data standards were not widely followed in the past, and so you can find latitudes and longitudes in a variety of different formats, just as one example, So if you want to actually analyze across any number of these samples, there's a lot of standardization that has to be done. So here's a better example from a different sample, and you can see here we do have some of that information. But again, this isn't the most easy to retrieve. And so this is a well-recognized problem, There's a well-recognized need to improve the situation. And efforts like the NOAA Omics Strategic Plan and the National Aquatic eDNA Strategy are providing roadmaps and resources. I also want to highlight that there are many ongoing efforts to improve microbiome data access. So you may be familiar with tools such as NMDC, JGI, MGRAST, and these databases and projects are doing great things. They're helping to make metadata or data in general more fair, which is an acronym widely used in this space to refer to findable, accessible, interoperable, and reusable data sets. They're also establishing standards for reporting metadata and for depositing data. They're also just in general encouraging public data sharing and collecting some of the rich metadata that's available. They're building great platforms and pipelines for data discovery and exploration. However, there are some downsides or areas we felt that we could improve upon, particularly for the Great Lakes. So for instance, NMDC doesn't currently support amplicon data or metabarcoding data, as you may be familiar with it, which represents the bulk of the data, existing data anyway, from aquatic systems. Some of these tools or efforts are also primarily forward-looking, such as NMDC. They're not going back to existing data sets and working to clean those up. Some of these tools are also, while very feature-rich, not the most intuitive or user-friendly. For instance, I would say that applies to JGI, or they rely on user submissions such as MGRAST. So GLAMR, or the Great Lakes Atlas of Multiomics Research Database, is made specifically for the Great Lakes. And due to the more tractable scale of samples, we're able to curate the data much further than some of these other efforts. So we've been working on curating previously deposited omics data sets, in addition to some of our own data sets. So we currently have nearly 5,000 metabarcoding samples, 1,500 shotgun metagenomes, and around 300 metatranscriptomes standardized into GLAMR. And you can see an accumulation of how these samples have been increasing over time. So really increasing, especially after the mid-2010s. So in addition to collecting these omics data sets, we've also been collecting Great Lakes relevant metadata. And because this database is specific to the Great Lakes, it also allows us to perform some targeted analyses that other platforms may not be interested in. So for instance, analyses that are really targeting Great Lakes relevant species or toxin biosynthesis genes, say relevant to the harmful algal blooms that some of the Great Lakes experience. So I mentioned that we are collecting the metadata. We're also extensively standardizing it. So this includes the complete sample metadata collected wherever possible, including sometimes going to the supplemental data associated with publications, tracking down environmental parameters, such as, say, right, the temperature where the sample was collected, also nutrient
Hi, Anders. I think it appears we lost audio. I did lose my internet connection for just a minute, but I'm back. Can you hear me? Anders?
Okay, can you hear me now?
Yes, I can hear you now.
Okay. I don't know
If
when
you
it cut out.
could, yeah, go back about a minute, definitely I think the prior slide here.
Okay. So start from the beginning here.
Yes, thank you. Sorry
Okay.
about that. I'm not sure if that was
Yeah,
my
no.
connection or yours.
It sounds like maybe something on my end. I switched my microphone, so hopefully things will work more smoothly from here on out. Okay, so I think I was mentioning that we collected all of the existing omics data sets from the Great Lakes. In addition to collecting the omics data sets, we also collected the complete sample metadata associated with the samples. So this included efforts to go back even to the publications associated with the data sets and dig into supplemental data to find all of the metadata that was available for the samples. So for instance, these metadata can include measurements of environmental parameters such as temperature, and then also measurements of nutrients collected alongside the omics samples. These also include parameters relevant to harmful algal blooms, where they were collected, such as phycocyanin or microcystin. We also keep track of any modifications or amendments that were made to samples so that we can make it easy to identify samples that are not representative of the natural environment. And then all of this metadata is standardized. So for instance, dates are put into the same format, latitude and longitude are standardized. And then all of the other parameters, wherever possible, follow widely adopted community standards, such as the MIX-S or minimum information about any X sequence standards, making this metadata interoperable with other platforms. We also leverage ontologies for location, environment, sample material, or other characteristics of the samples. And then finally, all of the units are actually converted to standardized units. And so all of this reduces friction and startup time, improving collaboration and facilitating future work such as modeling efforts. So as an overview of the organization of GLAMR, right? Individual studies generally are producing these data sets and collecting metadata, which they're depositing in other repositories such as NCBI. And as a side note, I'd just like to mention that we still encourage data to be deposited in repositories like this, right? We want all the raw data to end up in the same place. We're not trying to compete in any way for that space. So we pull down the data from NCBI and then actually process it through our own custom bioinformatics pipelines to produce standardized data products, allowing for analyses across any of these samples. And then later on, we'll touch on kind of where some of this ends up and the website that we've produced. So these standardized annotations are produced via our bioinformatics pipelines, which take sequencing reads, for instance, from quality control through determination of taxonomic composition, gene abundance, even the assembly and binning of metagenomes. So if you're not familiar with this process, right, in a shotgun metagenome, you're sequencing all of the DNA in a sample randomly. So if these are, for instance, genomes in the sample, in the sequencing, they get fragmented, and then they have to be assembled and use a process called binning to try and reconstruct the genomes as they existed in the original sample. So for instance, our pipelines have fully automated and standardized binning capabilities. All of this is implemented in the SnakeMake workflow manager, and these pipelines are available on GitHub for use by others. Oh, and I also mentioned that there's a wealth of annotation of these data sets as well. So for instance, for the MAGs, we can annotate genes associated with their metabolism or the biosynthesis of secondary metabolites. So here, for instance, you can see nitrogen metabolism associated with a particular MAG. To highlight the types of capabilities that GLAMR enables, I'd like to talk through some work done by Paul Denial, a master's student in the lab, and currently a bioinformatics specialist at the Cooperative Institute for Great Lakes Research. So there's been an increasing interest in a species of cyanobacteria in the Great Lakes called Dolichospermum, as it appears to be getting more abundant over time. It now causes small blooms in Lake Superior, and in the fall of 2022, there was a large Dolichospermum bloom in Lake Erie. Cyanobacteria are prolific producers of secondary metabolites of various classes, and a significant portion of our research outside of GLAMR focuses on those. So potent neurotoxins such as saxitoxin and anatoxin had previously been detected in Lake Erie, but it was unclear which organisms were responsible. So Paul leveraged some of the genome resolved methods implemented in the GLAMR pipelines and was able to discover a single metagenome assembled genome, or MAG, containing the biosynthetic gene cluster for the production of saxitoxin, which is a really potent neurotoxin. And he found that this MAG was very similar, right? This MAG here that has the saxitoxin biosynthesis gene cluster, so this Great Lakes strain, is very similar to other Lake Erie MAGs that did not contain this biosynthetic gene cluster. And the particular genes that were present suggest the production of saxitoxin, but thankfully not the production of a more potent congener, neosaxitoxin. So Paul also mapped reads from the metagenomic data sets to these MAGs to determine how the abundance of various Dolichospermum species has been changing over time. And you can see that many of the species, or the particular species that has the saxitoxin biosynthesis gene, is frequently observed earlier in the season. And the species that seems to be more abundant, particularly in the fall, and increasing in abundance, does not have the saxitoxin biosynthesis genes. So in addition, Paul discovered that the abundance of saxitoxin genes is correlated with temperature and negatively correlated with ammonium, also incorporating some data from qPCR monitoring. So that's an example of outputs and analysis capabilities enabled by GLAMR. And now I'd like to talk about data access and queries made possible via our website, greatlakesomics.org. So I'm going to actually hop over to the website and highlight some of its features. So on the GLAMR homepage, you can see an overview of all of the samples present, and you can, for instance, explore the map. Samples are colored based on their type, so amplicon or metabarcoding, metagenome, metatranscriptome. You can also find summaries of the available data sets. So for instance, if you are interested in the metagenomes in Lake Erie, or data sets containing metagenomes in Lake Erie, you could click on this and find a list of those studies. If you click on an individual study, you can then find more information about it. For instance, any publications associated with it, the samples associated with it, where applicable, links out to NCBI bioprojects, and then some other kind of sample level metadata about it. We can also go back to the GLAMR homepage and dive into samples, right? So kind of breaking them out of studies. So for instance, if you were interested in all of the metagenomes in Lake Erie, you could click on this link, and then by clicking on a particular sample, you can again see all of the metadata associated with this sample. So for instance, the name that the original collectors gave it, when it was collected, where it was collected. At this case, there are routine monitoring efforts conducted by NOAA GLAMR, so it includes the station where the sample was collected, right? And then some of these further characterizations, the temperature, measurements of some of the other characteristics that we described earlier. And this also includes references, again, for instance, to NCBI, right? So to be able to go back to the original source and find that data. I'll also highlight that file downloads are available, or you could browse a taxonomic summary that we've calculated for the sample based on the characterization of the reads that are present. And this is interactive, so you can explore and find the abundance of any organism that you're interested in. If you have a really targeted question, so for instance, let's say related to the work that I described Paul conducting earlier, you could search for a particular organism. So for instance, let's say Dolichospermum circinale, and you can then look for samples where this particular species has been observed. In this case, ranked by the abundance of the organism in those samples. If you were interested in looking for a particular gene, so let's say in this case, the SXTA gene associated with saxitoxin biosynthesis, you could search for that gene and you'll get several records. So these are UNIREF protein IDs with this particular name. And if you click on one of these, you can see the abundance of this particular gene. We see, in this case, it was observed in 64 samples. If you click on the abundance, you can then see which samples it was observed in, or even download a table of these abundances. So that is a brief overview of some of the functionality that we have implemented on the GLAMR site. I'd also like to highlight work that we conducted with GLOSS, the Great Lakes Observing System, to make some of our omics data processed through GLAMR available via their Seagull platform. So Seagull historically has been used for real-time data access on, for instance, data from buoys. But we worked with them to also accommodate omics data, and it's available on a layer in the Seagull platform here. So for instance, you can browse, let's just go back to the original omics layer. So you can browse samples. And in this case, we have only made a subset of the data that is particularly relevant to harmful algal blooms available. But you can, for instance, see the abundance of Anabina at a particular site, which is a species of cyanobacteria, and explore how its abundance has changed over time and the relative abundance of several organisms that are relevant to harmful algal blooms. In addition, you can see some of the environmental parameters collected simultaneously with these omics data sets and overlay them on the plots, right, to understand the relationships between some of these parameters and the harmful algal blooms. So as I mentioned, we worked with GLOS to make a subset of this available via the platform. I should also mention that on Siegel, you can also link out to GLAMR to find more information about the sample. In this case, I think that particular sample has not been made publicly available via GLAMR yet, even though some of the data was deposited in Siegel, which is why that link didn't work, but they generally do. So this was a great process, and we have a nice established data flow. So GLOS set up an API so that we can push new data sets to Siegel as they are available. I'd also like to mention that GLAMR has served as a home or a hub for omics training. So we have hosted weekly bioinformatics workshops for postdocs, students, and staff members. And these have largely centered around some of the methods implemented in GLAMR. And we've also hosted a workshop for Great Lakes researchers. This was held earlier this year and had pretty good attendance, representing many of the labs producing omics data sets from the region, and we hope to have more opportunities like that in the future. Looking forward, there's additional functionality that we are planning to implement in GLAMR. So for instance, one that is nearly ready to deploy is authenticated data access. There are cases where we don't want to make the data available immediately. So we've implemented methods to password protect data until its public release, allowing for analysis by relevant stakeholders. We'd also like to expand the data available via website queries. So for instance, metagenome assembled genomes like Paul used are not currently browsable or accessible, searchable via the website. So we'd like to make some of those available. In addition, we're always implementing additional omics processing methodologies. So keep an eye out for more annotations in the future. Additionally, we're working to improve the display of search and gene annotations and other instances like that. And we're also excited for other opportunities to integrate GLAMR with other services like we did with Siegel. So for instance, depositing biodiversity data into repositories such as GBIF or OBIS, supporting some of the biodiversity work that NOAA is very interested in. And then finally, we're interested in additional visualizations via the website. So before I close, I would just like to welcome all of you to contribute data to GLAMR. If you have Great Lakes relevant data sets, feel free to use the contact form on the website or email us at glamoromics@umich.edu. And finally, I would just like to acknowledge everyone who's made GLAMR possible, including the NOAA Omics program, NOAA GLURL, the Cooperative Institute for Great Lakes Research, Great Lakes researchers more generally, who've produced many of the data sets contained in GLAMR. It's a testament to the power of open data access and how that can really provide benefits for many years to come. I'd also like to thank the curation team who's helped to enter in and clean up and standardize much of the metadata in GLAMR. and highlight in particular Robert Hine, who's a bioinformatics specialist with the lab, who's been instrumental in implementing particularly the website in GLAMR and making sure that it's always up and running. So with that, I think we have time for questions. And again, thank you all for joining.
Yes, we do. And thank you, Anders, for the great presentation today on GLAMR. So we'll now move into the questions portion of the seminar. I have one, but we'll start off with one that came into the chat. So this one's by Kelly, who asks, can you explain more about the integration with GBIF or OBIS and NMDC? Specifically, what is the relationship to NCEI of the data within GLAMR?
Yeah, so at the moment, we have not integrated or pushed data from GLAMR to GBIF or OBIS. Those are things that we're interested in. So that would look like producing scripts to convert data into the Darwin core format that is widely used by those sites, or leveraging some existing work that I know has been done as part of the kind of broader NOAA Omics umbrella, particularly by some folks at AOML like Catherine Silliman and Luke Thompson. And then the relationship with NCEI at the moment, we mostly leverage NCEI to collect some of the metadata associated with omics data sets. But I think that there are future opportunities to deposit some of the data summaries inside NCEI and make them accessible that way.
Awesome. Thanks, Anders, for that clarification. We'll let others ask a few more questions as they come in. But I was wondering if you could speak to the quality control or analysis data process or for the data going into GLAMR. So what might that quality assurance look like as this data moves into GLAMR? How are those minimum requirements determined and integrated into GLAMR?
Yeah, that's a really good question. At the moment, we really do most of the curation in-house, which alleviates some of the concerns here, right, with user deposited data, although we certainly work with groups to collect that data and make it available to people working on curating data for GLAMR, or certainly reaching out and asking for clarifications where needed. In terms of the data itself, we have a variety of quality control steps implemented in the bioinformatics pipelines. So to account for some of the sort of technical artifacts that you can see. And then we also screen, for instance, for outliers in the data, metadata, or omics data. So it's a somewhat manual process to make sure that data is being held to a high standard. And we also certainly welcome feedback, right? If you, for instance, identify errors in your own data posted on GLAMR or other data sets, please reach out and we'd like to correct those.
Thanks. Thanks, Anders. I think we have a few more questions about particularly the NOAA Omics seminar series, and then we'll conclude. So we received one question about where we can get information about future seminars and or workshops. So I think when it comes to NOAA Omics, we have our seminar series is the third Wednesday of every month. And you can find details on that on the NOAA Omics landing page which you can find by Googling NOAA Omics, or there's a link in the chat. This question also asks, could we find more information about future workshops? So Anders, will there be any more presentations or workshops on GLAMR and developing this database into 2025, that is?
Yeah, there's not currently one on the calendar, but I would say if you're interested, please reach out via the email address that I shared earlier, so glamr-omics@umich.edu, or the contact link on the website, and we'll make sure to add you to the list.
Thanks. That one was from Emily, so there you make sure to write down glamr-omics@umich.edu. We did have one more question about the Omics, NOAA Omics workshops. I think is meant seminar series by this. And is it free, and how can we join? As mentioned before, yes, it is free. They're open to the public. You can find more on the NOAA Omics landing page linked in the chat, as well as NOAA Omics let me see if I can get you that URL. And I'll mention it for those joining on YouTube just a little bit later. Our landing page URL is oceanexplorer.noaa.gov/NOAA-omics.html, and that's particularly for those listening on YouTube that might not be able to get the link in the chat. Alrighty. With that, I don't think we have any more questions coming in. Anders, thank you very much for your great presentation on GLAMR today. We really appreciate you coming to speak at the seminar series. And thank you to all who joined. As mentioned, our seminars are held monthly, and we are establishing our schedule into 2025. You can find more on NOAA Omics on at the previously mentioned landing page. And you can also sign up for our listserv there to receive updates on future seminars. With that, Anders, thank you so much for joining us today, and I hope everybody has a great holidays.
Thank you so much.
Thanks. See you guys the next time.